Hello, everybody, and welcome to this GEN webinar. It's entitled GPS for Glycan Analysis, Routine Tools for Mapping Your Glycan Profile. Biopharmaceutical manufacturers are finding that the assessment of critical product quality attributes is increasingly a matter of evaluating post-translational modification. This is especially the case for glycosylation. But characterizing N-Glycan profiles has long proved difficult due to extensive labeling protocols and compromises in fluorescence and in the mass spectrometric sensitivities of different labels. Recently, however, a new technology has been introduced to address these challenges and to transform the N-Glycan profile analysis process. And that's what today's webinar is all about. I'm John Sterling, Editor-in-Chief of GEN, and I'm going to serve as the moderator. Our two webinar speakers will describe this new technology in great detail. They will also illustrate what it can do to significantly improve mapping glycan profiles. So why don't we meet them? Dr. Stefan M. Koza is Principal Applications Chemist at Waters Corporation. Stefan will talk about effective sample prep procedures for glycan analysis. Dr. Sean M. McCarthy, Senior Manager at Waters, will explore how routine monitoring can be carried out for the glycan analysis process more efficiently and economically. Now, after all the panelists have made their presentations, there will be a question and answer segment. Feel free to send in a question for our panelists at any time during the webinar. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. The panel will try to answer as many as possible. So, if everyone's ready, let's get going. Dr. Koza will be our lead-off presenter. Stefan, all yours.
Thank you, John. Hello. To start off this presentation, I'm going to be talking about the sample preparation aspect of N-Glycan profiling and some of the advances Waters has made in the past half a year or so in terms of developing a new system for both releasing as well as labeling and separating and analyzing N-Glycans. Before we get into the nuts and bolts of how this new kit works, what I'd like to do is first touch upon the glycosylation of biotherapeutics and why we care about what's happening with these molecules. Glycosylation of biotherapeutics can affect both the activity of molecules, the half-life of molecules, and as well as cause immunogenic responses in patients. And that's why they're important to monitor. A second reason they're important to monitor is that even if the changes don't affect the half-life or activity of the molecule, they're really sensitive, the profiles are really sensitive to changes in manufacturing conditions. We have to remember that N-link glycosylation and O-link glycosylation, for that matter, it's not a templated process like the transcription of DNA into proteins, but it is basically driven by the cell machinery. So slight changes in the health of cell culture can really cause significant changes in glycosylation profile. Within Waters, we like to think of the characterization of glycoproteins as really looking at multiple strategies that provide complementary information. And that's shown on this slide, where what we're trying to think about here is we're looking at anywhere from looking at an intact glycoprotein all the way to a monosaccharide released from the N-glycans or O-glycans of that protein, and everywhere in between. These bits of information can all add up to give us a good overall picture of what's going on with our glycoprotein. For today, what we're going to do is focus on the released glycan analysis. It's important to remember, as John mentioned, that this type of analysis in the past has been difficult or has produced glycans that don't give us mass spec or good fluorescence sensitivity. So what we focused on here at Waters and what I'm going to talk about with this kit is a simple and fast way to look at released N-Glycan analysis. Shown on this slide is a typical profile for the analysis of released N-Glycans, in which we want to obtain a helic profile. Generally, regardless of the chemistry used, what we're going to do is release the N-Glycans, and then label the N-glycans since they don't have any chromophores on them, purify them, and then analyze them by some separation. In this case, it's LC or LC-MS analysis, and then go through the data processing in order to understand our data. In the past, this conventional workflow has taken up to 5 hours to 2 days in order to just prep the samples. And what we're going to talk about now is a new technology that Waters has developed to basically make this process more straightforward, streamlined, and easy to carry out. Shown on this slide is a typical workflow for the labeling and release of N-glycans from a glycoprotein. Starting at the top, we have our glycoprotein, and we treat it with PNGSF, an enzyme that releases the N-glycan. It releases the N-glycan as a glycosylamine. Once that glycosylamine is released, it now has to cyclize into a reducing sugar, and then it can be labeled through a reductive amination process with 2AB. This process relies on numerous chemical conversions. It's laborious and time-consuming. And one of the more time-consuming aspects of this reductive amination labeling procedure is that it must be performed under anhydrous conditions, so no water can be present, which requires dry-down steps, which are time-consuming and add to the laboriousness of this process. On the left, if we took a rapid tagging approach, what we can do is once the N-glycan is released, we can then go ahead and label it as the glycosylamine by reacting with that amine group on the glycosylamine. How are we going to do that? Well, at Waters, we looked at currently available technology and decided that there really wasn't a really good way to label and analyze the glycosylamine. So we worked off of our previous experience with AccuFluor, which is, as many of you know, a reagent that's commonly used for labeling amino acids for amino acid analysis. And we decided, how can we make that better? So we took the rapid part of the amine labeling of AccuFluor, which is the carbamate group shown in green here. We added to that a fluorescence tag, which in this case is a quinoline fluorophore. And in the third part, we've added a tertiary amine charge tag. Without getting too clever, we decided to name this compound RapiFluor-MS because of the rapid tagging, the fluorescence, and the MS amine charge tag. What we were hoping to achieve with this is a compound that would allow for rapid tagging, efficient fluorescence signal, and because of that tertiary amine charge tag, give us good mass spec sensitivity. So this is the chemical process by which we go about tagging released N-glycans using RapiFluor-MS or RF-MS. In the upper left, we have our glycan with released as a glycosylamine, and we are going to add our RF-MS, and that will react with the glycosylamine and will wind up with a labeled glycan, and that's shown on the lower left. An important thing to remember about this label is that the mass shift that we expect for changes for this is 312.16 Daltons, and that's monoisotopic, from the glycosylamine, or if you're calculating from a reducing end sugar, it'll be 311.18 Daltons. And another important factor in this labeling is to remember that this carbamate, the NHS carbamate, will hydrolyze under water conditions. So it's hydrolyzing during the course of this reaction, but at a slower rate than the labeling is occurring. And in this case, it's a self-quenching reaction. How does this tag compare to currently available technologies? Shown in this slide is a comparison to instant AB. In the top left-hand corner, we have a comparison of the fluorescence and mass spec base peak intensity traces for RapiFluor-MS, and in the bottom left, we have the instant AB fluorescence, and mass spec. We can see that the fluorescence signals shown in the bar graphs on the right are pretty comparable, with the RapiFluor-MS maybe having twice the fluorescence intensity of the instant AB. But the major difference between these two tags is that when we compare the mass spec sensitivity, we can see that it's approximately 3 orders of magnitude greater sensitivity for the RapiFluor-MS labeled N-Glycan versus the instant AB labeled glycan. Now we want to compare the performance of our RapiFluor-MS label to the standard approach of reductively aminated 2AB labeled glycans. And that's shown in this slide. Again, we have the RapiFluor-MS fluorescence and base peak intensity traces in the upper left-hand corner. And now we have the 2AB labeled fluorescence and mass spec traces in the lower left-hand corner. What we can see is the fluorescence intensity is significantly greater for the RapiFluor-MS labeled glycans. It's approximately 10 or so times greater. Additionally, even though it's not as much of a change as we saw with the instant AB, the mass spec sensitivity difference is about 2 orders of magnitude greater for the RapiFluor-MS labeled glycans as it was for the 2AB labeled glycans. So we've now developed this wonderful new labeling reagent, and in order to use it to its fullest potential, we wanted to design a simple and streamlined method for labeling N-link glycans with this. So this is outlined in this slide. We start with our glycoprotein, and then we release our N-glycans using PNGSF. And we worked with New England Biolabs to come up with a solution for the PNGSF release in which we could get full release of our N-glycans in approximately 10 minutes. Following that, and without any dry down steps or any sample cleanup, we go ahead and directly label our glycans in a 5-minute reaction step. And then once our labeled glycans are ready, we then have to purify our glycans. And we do that using a Hillick microelution plate. And then, without an additional dry down step at any point, we can then go to direct analysis by Hillick. In all, for a few samples, the total sample prep time is approximately 30 minutes. I should point out that currently this reaction kit that is provided is for 96 samples. We've broken it down into 24 samples by 4 sets so that you can do 4 different analyses analyzing 24 samples at a time. We are currently working on developing and should soon have available a new kit, which we consider the light kit version of this kit, in which we will have the ability to analyze 24 samples in groups of 3. So the reagents will be supplied in a format so that you can analyze 8 samples at a time, and you will have enough reagent to do 3 sets of 8. And that's for customers who aren't doing really high throughput analysis. Let's look at the first step in this streamlined procedure. We're going to start with our glycoprotein on the left, and in the presence of 1% RapiJest, we're going to heat this sample for 3 minutes at 90 degrees. Then we're going to allow it to cool at room temperature for 3 minutes prior to adding our PNGSF. Once the PNGSF is added, we're going to incubate the sample for an additional 5 minutes at 50 degrees, and that's going to release our N-glycan. Shown in the bottom of this slide is an example for a MAB, where on the left we can see that we have a monoclonal antibody that's glycosylated, and on the right, after going through this procedure, we have the deglycosylated MAB mass being the predominant mass with greater than 99% release of the N-glycan. On this slide, what we want to do was look at the effectiveness of this procedure for other glycoproteins. And that was done by looking at a gel shift assay. And in this case, we're comparing it to a control in which we've done the denaturation with SDS and a long digest with PNGSF that has been shown to remove the majority of N-Glycans from glycoproteins. And what we can see based on the gel shift assays is that for these various glycoproteins, we get near full release of those as well. So the final part of this procedure is the cleanup step. We start with our labeled glycans, and now we have to remove it from the reaction byproducts, as well as the protein that was left over. And because proteins have primary means, that has also been labeled with our RapiFluor-MS reagent. To do this, we use a Hillock SPE cleanup step, where we collect our labeled glycans and throw away our byproducts. In developing this procedure, we wanted to make sure that it was robust, and that's shown in this slide, where we can see that if we don't do a cleanup, we have a large amount of reaction mixture components coming out and trailing into our main peak, but the profiles look comparable. And what we've determined is that with a 3 by 30 microliter water elution off of our SPE, that we can get about 75% recovery, and that still gives us a high enough concentration of sample that we can now directly analyze that. More water washes would lead to a higher recovery, but we thought that this was a good compromise in terms of analysis efficiency. To demonstrate that, we want to make sure that the SPE step did not speciate our sample, so we took our sample and treated it with SPE once and then a second time, and we compared our recoveries, and we can see based on the bar graph on the right that the recoveries are uniform for our different glycans going across our retention range for our Hillock separation. So in all, as a summary of this procedure, we can see that we have a half an hour sample preparation in which we get a really good yield of our N-Glycans and minimal amount of sample bias. In comparison to commercially available rapid N-Glycan preparation with 2AB, we get nearly double, or actually a little bit more than double the yield of N-Glycans. These derivatives are stable, and that's shown in this slide, where they've been allowed to sit at 10 degrees for over 3 days, and we get equivalent profiles. And that really covers the sample preparation and labeling. The next thing I want to cover is just a few of the other available items that can make N-Glycan analysis routine in your lab. The first thing we want to make sure that we provided was a RapiFluor-MS labeled glycan performance test standard, and that is currently available, and the separation of that based on analyzing by fluorescence and mass spec is shown in this slide. Additionally, as many of you are aware, the use of a dextran ladder as a calibration standard is particularly useful in glycan analysis. Shown here is the dextran ladder, in which we were able to label dextran with a version of our RapiFluor-MS that gave us equivalent fluorescence signal. And finally, in terms of accessories for your N-Glycan analysis, we want to make sure that making this mobile phase for the Hillock separation was easy and reproducible. So we developed a concentrated mobile phase for our elution buffer, in which it's very simply prepared by adding the contents of this concentrate to 1 liter of LCMS grade water. This concentrate is made from LCMS grade components so that we avoid the addition of adducts to our mass spec signal. And finally, I just wanted to cover that these methods are transferable. The helic separation that we've seen throughout this presentation has all been shown as being done on UPLC, but this separation is easily transferable and to a HPLC system using our 2.5 micron helic column. The only disadvantage here is that the runtime takes more than twice as long. And as a look into the future, soon to be shipping, we're developing a wider pore BEH amide column. It was originally developed for the separation of glycopeptides and glycoproteins, but we've observed with this column that when we look at highly extended structures, so your tri- and tetrasilated structures, that the resolution that can be gained for those structures is higher on this wide pore BEH amide column than is observed for the 130. So if your samples are highly silated, highly branched structures, this might be a column that you would consider using. So in summary, we've developed a preparation solution for labeling N-Glycans that can be done in as little as 30 minutes. This preparation, this label provides unprecedented sensitivity. The important aspect of this is that not only is the fluorescence sensitivity improved, but also the mass spec sensitivity is greatly improved, making characterization and analysis easier. And Sean will talk about that coming up in terms of routine analysis. And as well, we also supply all the other accessories that are needed in order to have a full workflow in your lab for the analysis of N-Glycans. And with that, I just want to remind you that questions can be asked by typing those in to the lower left-hand corner chat box. And now I want to hand off the presentation to Sean.
To begin, as Steve mentioned, I want to focus on some of the applications, in particular focusing on the use of the Acuity QDA for doing routine released N-Glycan analysis in conjunction with the RapiFluor-MS reagent. Sort of an outline of what I will be discussing. First, as I mentioned, I want to discuss the application of the QDA for released N-Glycan analysis and what the addition of mass confirmation can bring to your analyses. I then want to transition and discuss some of the chromatographic factors that can be adjusted to optimize helic glycan separations, in particular those separations of RapiFluor-MS labeled species. And finally, conclude with some extended applications that we've seen the QDA be useful for biotherapeutics. So to begin, as Steve mentioned, with RapiFluor-MS, we really sought to improve not only the sample preparation aspects of released N-Glycan analysis, but also to improve some of those aspects that we've commonly heard from our customers as being a challenge. So if you look here, we see a comparison of the fluorescence and MS response for a variety of different labels that are commonly used within the biotherapeutic environment. And if we pick a couple of these to do a comparison, we compare RapiFluor-MS versus Instant AB, which is another fast labeling reagent. With RapiFluor, we see about a 2x increase in fluorescence signal, but a 1,000-fold or 1,000x increase in MS response in comparison between the labels. If you look at RapiFluor-MS against what is a traditional label, 2-aminobenzimide or 2AB, see a 14x increase in fluorescence and a 160-fold improvement in the MS in comparing RapiFluor to 2AB. And finally, looking at procanamide, which is often used as a label when MS sensitivity is necessary. In comparison to RapiFluor-MS, RapiFluor yields, again, a 14x improvement in FLR response and a 3x improvement in MS response. So in comparison to other labels, we're not only getting a fast labeling, but also improving our ability to detect and monitor those structures. To highlight that even further, if you look here on this slide, we see a comparison of RapiFluor-MS labeled glycan structures, the spectra for two different structures, the FA2 and an FA2 with a bisecting GlycNAc, and compare that to the 2AB signal for those same species. And what we can see are two different things. Number one, if we look at the FA2 species, we see not only an improvement in MS response, but also the population of a much higher charge state. So where in 2AB we would see dominantly a +1 charge. With RapiFluor-MS, we see dominantly a +2. And then if we look at the structure on the bottom, again, we see a dramatic improvement in MS response, and again, a corresponding increase in the populated charge states, where now we have a +3 charge state populated. The box highlighted in green indicates the mass range of the acuity QDA. So the RapiFluor-MS label itself is enabling us to use something like an acuity QDA for routine monitoring by mass. So now I'd like to discuss, at least to some extent, the acuity QDA. So it's something that we introduced in October of 2013. And it really it's a revolutionary design that's meant to bring the power of mass data to chromatographers. As I'll show you in some of the slides that follow, the design of the instrument is very compact. We found it to be very robust and affordable. And it is an instrument that requires very little optimization. It's designed to essentially operate like many of the detectors that you would use, where you would turn them on and be able to collect meaningful data almost immediately. And it seamlessly integrates into our existing informatics packages such as Empower or in MassLinks. And in particular, looking at Empower, it can be installed and be used in a GMP-compliant environment, so enabling it to be used in qualified methods. So if you look at adding an acuity QDA to a system, you can see here highlighted in the green circle the actual footprint of the instrument. So it is very small, roughly the same size as a normal fluorescence or a PDA detector. As I mentioned, it's fully integratable into Empower CDS, and what we found is there's very little training required. Essentially, if you can operate an optical detector, you could operate a QDA mass detector. Importantly, for those people in environments like maybe in the United States or some places overseas and other geographies, it operates on either 110 or 220 volt, meaning that there's no special requirements for electrical needs within the lab like would be associated with other MS instruments. And the maintenance on the instrument is very minimal, and it's actually quite simple, as I'll show in a moment. So we look at first how to set up the QDA in order to collect mass data. You'll see here that the graphical user interface, or the GUI, for interacting with the acuity QDA is very similar to what you would have for a fluorescence or PDA detector, where in optical detectors you may set an excitation emission wavelength or a starting and an ending wavelength, say for a PDA. With the mass detector, you're defining a starting and an ending mass range. And in addition, there are some other parameters such as the cone voltage and capillary voltage that can be adjusted slightly. In our experience, there's very little need to adjust those values, as the instrument is very flat tuning, and it's been designed in that way to be pre-optimized and ready to deploy. If we look at some of the construction and layout of the instrument, as well as some of the other features, first, the instrument is designed to do an automated resolution and calibration check at every startup. So as a detector, it's meant to be shut off at the end of every day if you desire, and every time you power the instrument back on, it will ensure that it's working and still within specification. In addition, as I mentioned, the electrospray interface has been pre-optimized, and that gives you very flat tuning and very little need to adjust any of the parameters in order to collect meaningful mass data. And I show on the bottom the graphical display that you would see if you were looking at this instrument within our Acuity console, or the way that you would interact with the instrument. And part of showing this is to highlight the fact that while the instrument is meant to be a very simple-to-use detector, it does incorporate a lot of the technologies from our higher end and, say, more revolutionary mass spec instruments. And in particular, one of the areas is the step wave device. which is meant to improve the sensitivity. So even though the instrument is small and compact and easy to use, it takes advantage of a lot of the technologies we've incorporated in other technologies to enable users to collect meaningful and useful mass data on a regular and routine basis. I mentioned previously maintenance is quite simple, so there are really only two different aspects of the instrument that are meant to be a maintenance aspect that would be done by a user. The first is the sample aperture, which if you're familiar with mass spectrometry would be analogous to a sample cone. That's as simple to change as it would be a detector lamp. The aperture disc itself is meant to be disposable, so there's no cleaning of that, and it's priced as a consumable, so it's not expensive to replace. In addition, the capillary, which is essentially the sample probe for the instrument, comes as a pre-cut piece of tubing with the emitter tip fixed already, and that's as simple to change as changing any piece of tubing on an instrument. So no need to disassemble an entire instrument to change out the capillary. It's very simple to do. So in summary, the QDA, what we found is that it really is a revolutionary product that's enabling chromatographic users and those people who are not highly trained in MS itself to collect meaningful mass data on their own. And it provides mass information for each of your chromatographic peaks. In addition, we've also seen that it is very simple to deploy, and as I've shown, the construction of the instrument is very compact, which allows it to integrate very easily within existing instrument configurations, whether they be Acuity or Alliance. So now looking at the application of the Acuity QDA for some specific applications. What I show here is with routine monitoring of released N-Glycans. So these are released and labeled with RapiFluor-MS. And what we show here are three different types of samples. First is released glycans from an IgG, which represent more of the simple biantennary structures, which are fairly common. Released structures from RNase B, which represent some of those high manostructures, which Steve had referred to earlier, as potentially being a stability or process indicating type of structure that can a lot of times can show up in IgG profiles. And lastly, released glycans from bovine fetuin, which represent more of the complex, highly extended structures, highly silated structures. What's important to note here, on the top is our fluorescence trace, and on the bottom is the MS trace collected by the Acuity QDA. And you can see that each of the peaks in the fluorescence trace has a corresponding MS intensity on the QDA, highlighting the fact that the QDA can be used for release glycan analysis. And the benefits are that for each of your fluorescence peaks, you're now getting confirmatory MS information to more confidently and accurately assign each of the structures within that chromatogram. Another aspect of doing release glycan analysis, in addition to being able to detect the various forms of the release glycans and various structures, is to be able to detect those structures over a wide range of relative percent within a release glycan sample. So we show here the release glycan profile for an IgG. This is actually the reference standard that Steve had referred to earlier that we provide as part of the RapiFluor-MS labeling solution. We see here, we look at the most intense and the least intense glycan structures within this profile. We get very good mass data from the QDA for each of those structures, ranging from 20% relative peak area, and that's a fluorescence-based relative peak area, down to about 0.5% relative peak area. And we have examples where we've been able to see structures and get mass information down to about 0.1% relative peak area. So this highlights that our ability to see not only the most intense, but often the least intense, or the lower intensity structures with the QDA as well. And if we look at the structures that we would see within that release glycan profile, these are for IgG the IgG standard, you can see across all of the structures that will be contained within that, we're getting very good mass information and able to see those structures, whether they be in a +2 or a +3 charge state. And that's giving us the ability then to confirm those structures. Now, as Steve had mentioned, not everyone works specifically with IgG structures. So if we revisit a sample that we had seen just previously, which is the released glycans from bovine fetuin, again, we get very good fluorescence and MS information from the QDA, indicating that we can see those larger high-antennae structures. And if we look at the spectra for one of the larger structures within bovine fetuin, which is shown on this slide, you see this is a triantennary tetrasilated structure, and we're getting very good and meaningful mass information to allow us to confidently assign that very complex, highly silated structure with the QDA. And also highlighting that those larger extended structures do appear within the mass window when they're labeled with the RapiFluor-MS. And this is an example of something that would be very difficult to impossible to accomplish with any of the conventional labels that are available today. In addition, in looking at the application of the QDA, we see this applying much more towards a routine analysis where the profiles are probably largely known and have been characterized, and there's a desire to monitor them in downstream processes. And that's where the benefits of being able to incorporate the QDA into Empower are applicable. So shown here is an example of a chromatogram that was been processed within Empower. And you can see above each of the chromatographic peaks, we've been able to annotate that component with a specific name that is indicative of that glycan structure. And then the value, the numerical value above each of those peaks, represents the base peak intensity, or the most intense ion within the spectrum for the apex of each of those peaks. Again, adding confirmation that the structure that is assigned is the appropriate structure within that peak. In addition, the underlying mass information can be used to confirm that whether or not there are any coelutions or other species within any of those chromatographic peaks. In addition, as processes move downstream, there's often a desire to generate reports. So shown here is an example of a report where we have the relative peak area for each of the components. And then in a triplicate analysis, we're able to calculate standard deviations and things to monitor, maybe as part of process development, the changes in the different glycans that are present. Now, if we transition to look at another application, and this is an application where we've actually had quite a bit of interest and requests to develop some application towards this area, and that's developing a rapid method for doing release glycan analysis. So as we've been discussing in most of this webinar has been the use of a fairly lengthy but highly resolving chromatographic method for doing released N-Glycan analysis, and that's shown on the top. What we've done is we've scaled that method geometrically down to a very short 10-minute method. So that's 10 minutes from injection to the final re-equilibration of the column. And what you see is that while we still do get decent resolution from the majority of the species, there are particular areas where we're starting to lose resolution. The benefits are now, though, if we use and incorporate the use of the QDA to collect mass data, what we're able to do is monitor that very rapid method and select particular ions and SIRs, so those would be selected ion recording or collecting a specific chromatogram for each ion of interest. We're able to collect clean and meaningful mass data and chromatograms for each of the species that are of interest in our sample. And that's shown on the right, where we have selected ion chromatograms for the MAN5 species and the A2 species and so on. In each of those, we can see provides its own chromatographic trace. And even if the species are partially or completely co-eluting, we can discriminate between those two species. So in terms of doing a high throughput analysis, say maybe in a optimization of a bioreactor, a method such as this, coupled with the rapid labeling of the released N-Glycans, one could obtain from glycoprotein to actually interpretable result data within 40 minutes. In order for that to be of significant benefit, we needed to ensure that the response would be linear. So shown here on this slide are a series of spiked mannose into a pool of IgG-released glycans that have been labeled with RapiFluor-MS. Here you can see as the amount of mannose spiked into the sample increases, we get a correspondingly larger signal on the MS doing the selected ion recording. And that linear response allows us then to do some level of quantification and gives us assurance that the response that we would see would be linear, dependent on the amount of mannose or whatever species of interest were in that sample. And in addition, by using the SIR, we get unambiguous quantification, so no longer do we need to worry about potential coevolutions of structures. So we applied that and simulated essentially a bioreactor to look at how we can track the amount of, in this case, a mannose species in relation to another species, which would be desired within our bioreactor. And we could see is that as we increase the amount of mannose within that sample or within the bioreactor, we can see that the ratio of that mannose species versus the other is remaining relatively constant, which is what we would expect. So this gives us the ability then to selectively monitor particular structures within the separation. So with that, in the discussion of some of the applications, I want to discuss now some of the parameters that can be adjusted to optimize the separation of RapiFluor-MS labeled glycans. And throughout this, I'll be showing some of the data that we collected in conjunction with using the Acuity QDA, which gives us the ability to track particular components as we change parameters. So if we start first with temperature, and we look at the impact of temperature, we can see that as we adjust temperature, we really have very little impact on the selectivity of the separation, so all the components elute in the same order. We do see, as we increase temperature, that we improve our peak capacity, and we also improve the resolution. In this case, picking just 3 critical pairs within that separation, where we're getting varying amounts of either improvement or maybe even loss of resolution. So temperature certainly plays a role in the resolution of the separation. If we look at 2 specific structures, looking at the mannose and the FA1G1 structure shown here, we can see that as we decrease the temperature of our separation, we're actually gaining some of the resolution of the mannose species against that other component. So certainly temperature, depending on the structures you're looking at, would be something to consider to potentially improve that resolution. Another factor that's commonly looked at would be flow rate. Again, as we saw with temperature, there's very little change in selectivity. And as we increase the flow rate, we see a corresponding decrease in the peak capacity of the separation and also largely a decrease in resolution. So moving to lower flow rates could provide better peak capacity and better resolution. If you look at the impact of flow rate on 2 different critical pairs, one being the isomers of the FA2G1, you can see at a lower flow rate we're getting a better resolution of those 2 isomers. And with decreasing flow rate, we're slightly improving the resolution of mannose, MAN5, from the FA1G1. And this is a good opportunity to point out the use of the QDA. So each of the traces shown on this slide are created by interrogating the MS data. So you can see in particular with the mannose versus FA1G1 at 500 microliters a minute in the lower right-hand corner, without mass information, there would be no indication as to that coelution, as the structures are almost perfectly coeluting. If you look at the gradient slope of the separation, you can see that there is some minor change that we see in the selectivity of the separation as we change the gradient slope. But largely we see the larger impacts on the peak capacity of the separation as well as the resolution, where more shallow gradient slopes yield greater peak capacity and also greater resolution of the structures. If we look at a particular part of our chromatogram, again, now focusing on the mannose and the FA1G1, we can see that decreasing the gradient slope down to 0.2% B per minute almost yields baseline separation of those. So certainly looking at gradient slope would be something to consider as you're trying to improve the separation of a critical pair within your chromatogram. Moving on to looking at the impact of the ion pairing. So this would be the concentration of the ion pairing reagent within your mobile phase. You can see that to a large extent, the majority of the peak capacity, the selectivity, as well as the resolution remain fairly constant, although there is some impact on the resolution of the separation as you change ion pairing. But largely what we see is the chromatographic profile starts to migrate towards a slightly earlier elution as the ion pairing concentration goes up. And that's highlighted here where we see monitoring three different structures, the mannose, MAN5, FA1G1, and the A2G1 species at 10, 25, and 75 millimolar, where we see an improvement in resolution as we go to a higher ion pairing strength, while all of the components are eluting from the column slightly earlier. So the ion pairing concentration can play a role certainly in improving the resolution of your separation. Finally, if we look at column length, it probably doesn't come as a great surprise that as the column length increases and you scale the gradient proportionally with the column length, that you get an improvement of resolution, which is to be expected. And the incremental change in resolution is directly correlated to the length of the chromatographic column. As a reference, the majority of the data that I discussed and that Steve had shown was on a 2.1 by 150 column to give the highest resolution. The high throughput method I discussed previously was run at 2.1 by 50. But again, using the QDA in that aspect, we're able to overcome some of the challenges of the loss in resolution by selectively monitoring species of interest. So with that, I want to move slightly away from specifically looking at released N-Glycan analysis of the QDA and sort of give you a little bit of information on some other applications that we've seen the QDA be useful for within biopharmaceuticals. And as I'll mention later, we have an upcoming webcast that will describe a lot more in detail some of these applications. So first, I mentioned a large number of the laboratories doing released N-Glycan analysis. There's also a fair amount of peptide work being done. So here are two total ion chromatograms collected from the Acuity QDA. This is looking at a trastuzumab digest. Whether using a TFA gradient, which is probably the most common, or using a formic acid, a gradient with formic acid as the ion pairing reagent, we get very good signal from the QDA for all of the peptides within that sample. So the QDA is applicable for peptides. Even, I don't show the data here, we've seen peptides up to 7,000 molecular weight and even greater going up to some of the subunits. Another application area in which we've seen and heard a lot of questions about is in the area of excipient analysis, particularly in looking at polysorbate 80. So shown on this slide is the analysis of polysorbate 80, first looking at ELSD, or evaporative light scattering, to get quantitative information about the amount of detergent within a sample. So you can see we get a very linear response dependent on the amount of polysorbate within the sample. So we're able to quantify how much of the detergent is in there. In addition, by setting the system up as a split flow where some of the flow goes to the ELSD and the remainder of the flow goes to the Acuity QDA, for each of the points across that calibration curve, we can get meaningful mass information to understand the heterogeneity of the detergent. So that allows us to not only address the heterogeneity of the detergent within the formulation, but also potentially to assess raw material heterogeneity and be useful for things like stability testing to look at the impact of the detergent. Has the mass profile or the identity of the polysorbate changed, not only its concentration? What we've also seen is that the general applicability is across all of the non-ionic surfactants, so across Bridge or Triton or other commonly used detergents, so polysorbate 20. So it's not just applicable for polysorbate, but other detergents that may be used within your labs. Finally, another area where we've heard a lot of conversation is the applicability of the Acuity QDA for doing oligonucleotide analysis. So shown here are representative spectra as well as the chromatogram for the separation of oligonucleotides and their detection. What we see are a number of charge states that represent the spectra for each of the oligonucleotides. In this example, the spectra on the top represent oligonucleotides ranging from 15 up to 35 oligonucleotides in length. So there's no limitation necessarily that we've seen for the commonly investigated oligonucleotides, and the QDA is certainly applicable for monitoring those as well. So in summary, throughout the course of this webinar, what we hope we've shown is that the GlycoWorks RapiFluor-MS Glycan sample preparation kit can yield a 16x faster sample preparation time compared to conventional methods. So this will be going from maybe 5 hours to 2-day preparation and reducing that time down to about 30 minutes. We can see the RapiFluor-MS labeled glycans with the Acuity QDA. And importantly, that QDA is applicable in a number of the GMP compliant workflows in which we would see that instrument being applied. And by introducing the use of the QDA for glycan analysis, it gives you greater selectivity and the ability to run faster methods and get routine mass confirmation for the more routine types of biopharmaceutical assays that you would execute in your laboratories. So with that, I want to point you to some additional information. So if you want to learn some more about the development and more the science behind RapiFluor-MS and our evaluation of that technology, we recently had published an analytical chemistry paper where we discussed in great detail some of those aspects that were often asked about RapiFluor-MS and its applicability. In addition, if you go to www.waters.com/glycans, you can see the number of application notes and other supporting information as they relate to RapiFluor-MS. In addition, we have a webinar that we recorded and is available from earlier this year, which was from March, where we discussed in more detail the sample preparation aspects of RapiFluor-MS. And finally, upcoming in July will be another Gen webinar where we'll be discussing the applicability of the QDA for a wide range of biopharmaceutical applications, if you're interested in that. Finally, I'd like to acknowledge some of the people that have participated in the generation of some of the data you've seen today. Matt Lauber, Robert Birdsall, Owen Cosgrave, In-Ching Yu, and Daryl Bushmish, who all were involved in either the development or the evaluation of the data that you've seen here. So with that, we'll open the floor to any remaining questions. Thank you very much.
And Sean, thank you and Steph for those very detailed presentations on this very novel technological approach to improving glycan analysis. You both provided some extremely important information that has definitely resonated with our audience, and I can tell that because we've got a number of very interesting questions. So why don't we take the first one to begin our Q&A segment? Okay, Sean, first question. How does the chromatography of Fluor-MS compare to AA and AB labeling? Can the current databases be used, or do new databases have to be established?
The chromatography, as some of the examples we had shown, they agree very well with traditional 2AA and 2AB chromatograms. In terms of looking at those, the GU values that would be derived from a 2AA or a 2AB, they do vary slightly. And for that reason, we're in the process now of working with NIBRT in Ireland on the creation of a specific GU library that's focused primarily on RapiFluor-MS, and that should be coming. There'll be a version that should be available by the end of the year.
And do the Hillock conditions lead to any MS ionization suppression?
So the sensitivity and the conditions that we use for the chromatography do yield very good chromatography as well as good MS ionization. For me to say that there's no ion suppression, I don't think that would be completely accurate, but it is minimal ion suppression, and the improvements on MS sensitivity by using RF-MS are substantial enough that you still maintain very good ionization across the variety of structures.
Thank you. And how much starting material is needed for the reliable release of N-Glycans using this kit? Usually more material is needed. Well, why don't you answer that?
So the amount of material, the protocol that we've written has specifies an amount of 15 micrograms, and of the final released N-Glycan amount from that 15 micrograms of protein, using a high-end MS, there's way more than enough material for doing multiple injections. And for the QDA analysis, you could probably do about 4 injections of that same material. So there should be no need to scale up in order to get adequate MS information.
And many folks may not have the resources to purchase a mass spec. So do you have pre-labeled standard glycans that can be used for identification of the peaks on a glyco profile?
Yes. So we do have actually 2 different labeled glycans, so to speak, standards. So one is a pooled IgG labeled glycan standard that we sell that can be used, and those peaks have been identified. In addition, we also have a RapiFluor-MS labeled dextran ladder, which could be used for assigning GU values. I would caution that that there needs to be some confirmation that the structure is being assigned from the standard versus a sample. Then there would need to be some level of confirmation, I would think. But yes, we do have a labeled standard that can be used as sort of a preliminary guide of what structures are in your sample.
Another question, does the entire flow go to the QDs, or does it require a branch that needs to be tuned?
Yes, the entire the entire flow of the separation goes from the column through the FLR detector and finally into the QDA. There's no need to split the flow or to have any makeup flow whatsoever.
And are issues with capillary clogging common with this instrument? That is, how often does the capillary need to be changed?
So that's a good question. What we have seen in our experience, we've been running this application now for probably about a year. There's no massive problems with capillary clogging. And in general, across the variety of applications we've used the QDA for, capillary clogging does not seem to be a large issue. That's primarily due to the fact that the eluents are not high salt containing. So I would say no, it's not a large issue. In terms of how often it's replaced, it's hard to say how often it would be replaced because outside of it getting clogged, there's really very little need to change it. So likely to PM is when it would be changed. And the clogging issue, it's really comes down to care and use of the instrument. And how do the SPE recoveries vary based on the type of glycan? So that's something we've assessed, and Steve actually had a slide in his portion of the presentation that described that, as well as some information in our previous webinar. What we found in looking at the SPE results, if we do a single SPE pass and then follow it up and do an analysis of that sample and follow it up with a second SPE, the relative abundance of each of the glycan structures remains constant. So that leads us to believe there's no bias in particular structures in using the SPE. We're getting an unbiased or a fair representation of all the structures that are present in that sample without any specific loss of any structure individually.
Well, thank you, Sean, and that brings us to the end of our program. But please note that this webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to your colleagues and friends, which we highly recommend. I want to say thanks again to the panel for their outstanding presentations, and thank you to the audience for your attention and for your extremely thoughtful and pertinent questions about various topics brought up during the webinar. And thank you to Waters, whose support made this webinar possible. Bye for now.